Okay, so let's quickly have a look at these guys. So we only had to do 2, 6, 7, 10, and 11. Right? Number 2. What are we looking at for number 2? That is the smooth ER. Okay, number 1 was pointing at the rough ER. Number 2 is pointing at the smooth ER. And as with the last time, I will accept as the function any one of the following things. Repairs membranes, makes lipids, makes hormones, breaks down complex carbohydrates, and detoxifies poisons. If they have any one of those, they can have the mark for the function. Okay? Number 6. We're looking at the little dots. Sometimes they're attached to the rough ER, that's what makes it look rough, and sometimes they just float freely in the cytoplasm. What are they? They are ribosomes. And what do ribosomes do? They make proteins, synthesize proteins, yeah, okay? Number 7, this big thing right here. That is the Golgi apparatus. And what does it do? It packages wastes, right? Excretes wastes, sends wastes to the membrane, begins exocytosis. Anything along those lines would be fine. That's fine. That's what exocytosis is. Okay, number 10. Okay, we're looking at these two bubbles which both have stuff in them. And this, since this is an animal cell, these would be lysosomes. And lysosomes do what? They digest food. All right, and number 11, same thing. It's only in an animal cell, because only animal cells need these, can we? These are centrioles. And what do centrioles do? Yeah, that's good enough. If you said helps with cell division, that's really what they do. If you were more specific and you said put out fibers that grab chromosomes or haul DNA to either side of the cell, or something more complex than that about, you know, moving DNA or cell division, that's fine. Perfect. Yep. Perfect. Yep. Pulls chromosomes. Yep. Okay. Any other ones there that maybe not sure of? All right. So it's out of 10. Okay, because you only did 5, so give them a mark out of 10. There's 1 for the name, 1 for the function. Let them see it, put it in the box real quick, and then back to your desk. Okay, so the purpose of the cell anatomy lab is going to work a lot like the microscopy lab. We're going to go in there, and we're going to look at a few slides. Okay, but the focus is different here. And I don't mean that as a pun because we're using microscopes, because that would be a terrible joke. Okay, what I mean is this. When we had the microscope lab, we were mostly just working on, can I get stuff focused? Things look different. That's kind of cool, right? But in this lab, we're seriously looking at the internal structure of the cells we view. We want to look for common structures. We already know that there's a bunch of structures in common between plant cells and animal cells and all that kind of stuff. But what we want to find out is which ones are the most important. Okay? Because what the purpose of this lab is to investigate which parts of the cell are necessary to carry out the second point of the cell theory. Anyone remember what the second point of the cell theory was? Okay, first point was all things are made up of cells. That was the third point. Okay, the second point says, what do cells do? They carry something out. Okay, they do carry out jobs. The wording of the cell theory was this: they carry out the basic functions of the organism. Okay, so what we want to look at is what structures are in all the cells we look at. If they're in every single cell, then they must be responsible for carrying out some basic functions. Gee, that sounded kind of like a hypothesis. Had if, and then in it. Scary. Okay, see how important it is to pay attention? All right, so that's what we're looking for. Okay, we want to find out which of these things are in all of the cells, because if they are, there's got to be a reason for that. Okay, so problem, cells have to carry out the basic functions of an organism. We know that, okay, in order to survive. Thus, they must have structures within their cells that carry out these basic functions. We have to restate that in the form of a question. I think that would be pretty easy, starting with maybe the word which, okay, and not the wicked kind, the other kind that means either or, okay. For the design, manipulated, responding, controlled variables. the manipulated variable here, guys, is what we're going to make that's different. Okay? Are we going to look at different kinds of cells today? Yeah, we are. All right? They're all what we call eukaryotic, which was a term that was in your podcast on Thursday. Okay? Eukaryotic cells are cells like we have. They have organelles in them, a nucleus and mitochondria and all that. Okay? All the cells we look at today will be eukaryotic cells. Okay? That's something that they'll have in common, or that we might be controlling. Okay? But they're from different organisms. Okay? And that's the thing we want to confirm. Okay? So we're taking cells from different organisms, different kinds of organisms. Okay? What we're looking for has to do with this. It has to do with the problem. Okay? So our responding variable has to do with what we're hoping will be there. Okay? In our hypothesis, okay? Generate a hypothesis about which organelles will be present in cells and visible under the light microscope. For this, I think the if part needs to talk about the second point of the cell theory. Okay? It needs to talk about the problem. Okay? And the problem is this stuff about cells having to carry out basic functions. If they have to do that, whoops. Okay? The and part talks about what we're doing in this experiment. And the then part is, what am I going to see? Okay? You might even want to list what structures you think will be present in all of them. Okay? But most importantly, which ones you're actually going to be able to see. Okay? Because are you going to be able to see ribosomes? No. Okay? If you label ribosomes on your lab drawing, I'm going to call you know what on that. Okay? There's no way you see those. All right? In fact, you probably won't see a lot of the things that are on your cell diagram, but there are some things you will see. Okay? I've had people in this lab many, many times, okay, be able to see more than just the nucleus, the nuclear membrane, and the cell membrane. Yes, you're certainly going to see those things. Okay? But I have had people who are able to find the rough ER. Okay? Because if you looked at that diagram we just labeled, the rough ER is always attached to what? To the nucleus. Okay? It kind of looks like the nucleus has a beard most of the time under the microscope. Okay? Sometimes the beard is on top, but yeah, you kind of get the idea. Okay? They usually can see a Golgi apparatus. I've had lots of people able to see those. Okay? And lastly, okay, quite often in the plant cell, you can find the chloroplasts. They're going to be small, oval, kind of grayish green structures. Okay? You should be able to see those things for sure, so make sure you're looking for those. I have had people who've seen even more than that. Okay? But realistically, you should be able to see those things. Smooth ER is really hard to see because it's pretty much transparent. It's hard to tell from the cytoplasm. Okay? So I doubt you will see that. All right. So the slides we're going to be looking at. Okay? We're going to be looking at a monocot dicot cross section. Okay? What that means is this. We're looking at a leaf. Okay? We're looking at a leaf that's been cut this way. All right? So they've snipped it like this, and we're looking at it straight on. All right? So in its thinnest dimension, that way we can see the layers of the leaf. All right? So it's going to look something like this. All right? So we'll be looking at it like this. This up here is the top of the leaf, and this is the bottom. All right? We want to look at these cells in this layer. We talked about this, okay, on Tuesday. Okay, when we talked about the plant, and I showed you a picture of the leaf. This is what we call the palisade layer. Okay, and that's where the main kind of generalized shaped plant cells are located. All right, and you can see in here already, there's quite a bit of stuff inside of these cells. Right, so you should be able to see those fairly easily and identify some pretty good structures. How many of those cells do you need to draw for your lab drawing, or how many, sorry, do you need to label? Just one. Okay, so when you're doing your lab drawing and you snap the picture there, just use one of them as a label, unless you see something in another one that you didn't see in another. Okay, we're also going to be looking at this. This is going to be our animal cell. Okay, it's the stuff I told you about the other day. It's called stratified squamous epithelium. It's the skin that you can see when you dust your knuckle, okay, and it peels down to that red skin that hurts really bad but doesn't bleed very much. Okay. Okay, that's what you're going to be looking at. Okay, and you can see, okay, there's lots and lots of cells in there. Okay, each one of these things here, okay, like this here, that's a cell. And you can see inside there's a pink dot in the middle of it. That's the nucleus, right? You're going to have to zoom right in on that as much as you can, okay, to see all of the structures, but you can see quite a few things in there. Okay, we're going to be looking at blood cells, not because we want to compare them to anything else, because actually in a blood cell you can't see anything. The nature of their membrane makes them kind of opaque, and you actually can't see any of the structures inside. The only reason we're looking at it is because people always ask to look at it. Okay, people wanted to lance their fingers and look at things. We can't do that. Okay, we have these sealed up, prepared slides of human blood for you to look at, but it's actually not going to be part of your analysis. You're not going to include it, okay, as one of the ones we've used. And the last one we're going to do is one of your own cells. It's a cheek cell. Right. So what you're going to do is you're going to take a Q-tip and you're going to very gently swab the inside of your cheek, just like they do on CSI. Okay, this is what they gather when they do that on CSI. They strip some cheek cells off the inside of your mouth, and those contain DNA and all that kind of good stuff. Okay, so you'll do a wet mount of those. So when you've kind of smeared that thing around in your mouth a bit, okay, you're going to smear it onto the slide. And when you do that, okay, if you watch my marker here, as you smear it on the slide, you also want to twist it back and forth, so you get all the sides smearing onto your slide. Add a drop of water, put a cover slip on like you did with the letter E, okay, and you will see something like this. All right, cheek cells are these kind of grayish circles here, right, and you should be able to get a fair amount of those. Now, regardless of how well you rinsed and brushed your teeth this morning, there's still going to be chunks of food that come out of here. They're microscopic, but they're there. They look really big under the microscope. If something's really brightly colored and really dark and really big, it's not a cheek cell. It's something you ate. Okay? Don't take a picture and label that, because there won't be any structures in there that you need. Okay? It'll be kind of cool to see, but okay? Generally, you don't want to be looking at that. Most of the time, it's something cereal, like, okay, like a husk from a piece of grain or something like that, especially if you had shredded wheat. Okay? So yeah, things like that. This is what we're looking for here. Does that give everyone a pretty good idea what you're looking for? Okay. So since we're going to take the Chromebooks across the hall with us, okay, there's only one analysis question here, guys. Okay, but I'm going to want a pretty thorough explanation of it. All right, so we're saying like 3 sentences for sure. Okay, probably more than that to explain all of this stuff. It says, based on your knowledge of cell structure and your observations of different cells, not including the red blood cell, okay, in this lab, what organelles are most important for a cell to function, and which ones are specific to a certain mode of nutrition? Remember that we talked about chemoheterotrophs and photoautotrophs. Okay, obviously we're looking at one organism that's a chemoheterotroph, one organism that's a photoautotroph. They are going to have some differences. You'll need to talk a little bit about those. Okay, explain your reasoning. That's your only analysis question, so it's kind of like a short paragraph answer. Okay, then conclusion and evaluation, same as always. All right, questions? Okay, the biolab is open. Okay, you can head across there right now.